So the DNA can be damaged in a number of different ways by the radiation. Um, Destruction or changes of a base is a type of mutation, regardless of the severity of the consequences. As it is the sequence of all the bases in the DNA that store and transmit the genetic information. A change in this genetic information is, by definition, a mutation. Recall that the bases, though, on opposite strands must be complementary. Adenine pairs with thymine, and guanine pairs with cytosine. Other ways in which radiation can damage DNA involves single strand breaks. Breaks in just one of the strands of the DNA molecules have been measured experimentally in cells, but have been found to be relatively inconsequential in the terms of cell death. Although single strand breaks do occur as a result of irradiation, these are most likely efficiently repaired with little, if any, long-term consequences to the cell. In other words, they all repaired readily using the opposite strand as the template. There is a possibility that if the repair is incorrect, it can result in a mutation, though. When we think about more consequential damage, we're talking about double strand breaks. So these are much more damaging to the cell. This occurs if both strands break opposite one another, or if the single strand breaks are separated by only a few base pairs. Then essentially the chromatin snaps into two pieces, which is the same as a double break. And because the template is lost, this is not so easily repaired. Now, this will most likely cause the cells to die, although it could cause carcinogenesis, the initiation of cancer formation or mutations. For low LET radiation, the breakage of the DNA chain could be like 20 times less likely than a single strand break alone. And so the double strand breaks are more often formed by single tracks of high LET radiation. Another effect that can happen is cross-linking. In addition to breakages of the DNA strands, radiation can cause cross-links. An interstrand cross-link can be formed between two regions of the same DNA strand. Or an interstrand cross-link can be produced between two complementary DNA strands, or even between completely different DNA molecules. It's not clear how important this is in terms of the biological effects, and it is believed that this damage is probably repaired. Chromosomes produced by radiation can be observed microscopically. When the chromosome is broken, you can have two or more chromosomal fragments, each having a broken end. These broken ends have the important capability of being able to join with other broken ends, usually with the first broken end encountered, possibly forming new chromosomes. These new chromosomes may or may not appear structurally different from the chromosome prior to irradiation. The cell cycle phase determines a cell's relative radiosensitivity, with cells being most radiosensitive in the G2 to M phase, less sensitive in the G1 phase, and least sensitive during the latter part of the S phase. During the S phase of the cell cycle, the chromosome basically lays down an exact duplicate of itself, and then these two like chromosomes are termed sister chromatids. Chromatic aberrations are those produced in individual chromatids when irradiated after DNA synthesis, because only one chromatid has been damaged, and only one daughter cell will be affected. Chromosome aberrations, on the other hand, are produced when irradiation occurs before DNA synthesis. A resultant break will be replicated if repair is not completed before the initiation of DNA synthesis, and both chromatids will exhibit the break, and both daughter cells will inherit a damaged chromatid. Let's consider the outcome for this chromosome damage.